thanks for coming to the Cancer Center Symposium. It's my great pleasure to introduce an actual evolutionary biologist. So Ed Choung comes to us from University of Colorado in Boulder. He's the Marvin Carruthers professor or assistant professor there at the Biofrontiers Institute. He did training starting as an undergraduate, and I normally don't mention undergraduate except that his advisor was Hopi Hoekstra, the famous evolutionary biologist. And then he went and did after a PhD at Stanford, he then went on to do a lot of what he has continued on as an assistant professor, work with transposable elements and how transposable elements can be used as fodder for evolution. In this case, it's actually quite medically relevant because it's fodder for evolution within the interferon signaling pathway. And so by understanding how this diversity of phenotypes can be created by these transposable elements and creating new sequence elements that can be selected upon, we can better understand, evolution, but also how these different systems that regulate such important processes like inflammation have been evolved. And I think this is reminiscent of what Ian Malcolm said in Jurassic Park, life will find a way. And I think we're going to learn about one way that life found a way. So, Ed, and I'll turn this off because...
All right, is this working? Sounds like it's working. All right, well thanks, James, for the introduction, and it's really a pleasure to be here. I'm really thankful for the opportunity to present my work. And as James said, my formal training is really in evolutionary genetics, and cancer is kind of a new direction for my lab, which I started about 1.5 years ago. So I'm really eager to get any comments or feedback or questions that real cancer biologists may have. All right, so I'm going to talk about endogenous retroviruses and their influence on human biology and evolution. And so first I want to motivate this by talking about transcriptional regulatory elements, which is really the focus of our lab. Non-coding regulatory elements have increasingly become appreciated to drive diversity between species, between individuals, and also differences in disease susceptibility. And so when we think about where do these regulatory elements come from in our genome, it turns out there's some pretty interesting mechanisms. So first you can imagine that a regulatory element is essentially a collection of regulatory motifs and binding sites for transcription factors, and these may spontaneously arise through mutation. And so this is probably the predominant mechanism by which the non-coding landscape in our genome is shaped. But what I'm going to focus on is a different, potentially more interesting mechanism, and that's the replication of genetic parasites known as transposons, and specifically today I'll talk about endogenous retroviruses. So I want to give a couple slides of background on transposons. So first of all, if we think about the entirety of our genome, 3 billion base pairs, only about 2% of those 3 billion letters encode for the protein coding genes that most biologists know and love. But at least 50% and probably more of that genomic space is actually taken up from fragments of transposable elements, which are essentially pieces of DNA that don't do anything good for the host, but encode the ability to copy and paste themselves throughout the genome. So they're genetic parasites. They've been coined as the selfish gene from this old famous book by Richard Dawkins. And when we say transposons, it's actually a hodgepodge of different kinds of elements. They're not necessarily related. You've probably heard of line elements and sign elements, DNA transposons. I won't really get into the details of all of them, but they basically have structures like this where they're relatively small, up to 10 kb elements that contain regulatory sequences and proteins whose function it is to basically help replicate the transposon. So I want to go through a little bit of an example here. When we talk about endogenous retroviruses, all of them actually originate from real infectious retroviruses that happen to infect the germline cell long ago, and whatever individual grew up from that infected sperm or egg had this integrated provirus in them. So that seems like a pretty rare event, but it actually happens quite often, and there are many of these elements in our genome. So a provirus might look like this, where it's a retroviral genome in the cell genome. Those proviruses will hijack cellular transcription machinery and become transcribed, form transposon RNA, and oftentimes these RNAs will become translated into transposon proteins, including reverse transcriptase, integrases, polymerases. Eventually, those RNAs are reverse transcribed to cDNA, which are then reintegrated back to a new semi-random location in the genome. And I should say that, this is obviously a very powerful form of mutagenesis, and there are many instances of human diseases where a new transposon has jumped into an important gene, caused disease. By and large, though, most new insertions are kind of in the middle of nowhere and don't do anything. So we mostly think of a lot of these insertions as neutral or nearly neutral with respect to host fitness. And when you have something that's a neutral piece of DNA in a genome as large as ours, there's always a random chance that it'll just fix in the population, whether or not it's beneficial or not. So this has happened a lot. And we like to think of this as a copy and paste mechanism. So a lot of times people think of our code or our genome as kind of our code or the source code for our development. And in a way, a transposon is really like a computer virus or a bug that is just copying itself all around that code. I just want to give a view of what that looks like in a genome browser. So my lab spends a lot of time looking at genomic data. So here I'm just showing a locus, about 500 kb region, which has a couple of genes. And the entirety of our genome has about 50,000 protein coding or non-coding genes. But there's about 100 times more than that of transposon remnants. So that's what the repeat mask you're tracking in your genome browser is, all these pre-computed maps of where transposons are in our genome. I should say, thankfully, 99.9% of the transposon sequences in our genome are replication incompetent, which means that they've either degraded, suffered point mutations, or epigenetically silenced such that they can no longer replicate. So by and large, these are under epigenetic control. But of course, some always leak through, and that's going to be the topic of my talk. Generally, my lab looks at transposons as a double-edged sword in our evolution. You can imagine these are parasites, these are mutagens, so they're going to cause disease, and that definitely happens. But at the same time, because they're continuously providing new material into our genomes, they can be fodder or substrate for evolutionary novelties. So I want to focus a little bit on the evolution part in the first half of this talk, and then I'll go into what we're working on in context of disease. So as these endogenous retroviruses invade, they may, when they fix into the genome, they're essentially providing a new gene or a new regulatory element into that genome. And it's certainly, over the past few decades, there's been a number of examples where these retrovirally derived proteins have now become essential for proper human development. So one example is a gene that's expressed in the placenta. It's called syncytin. And the syncytin gene actually derives from the envelope protein of a retrovirus. And it's very highly expressed in the placenta. And it turns out that it's required for the formation of this special tissue barrier called the syncytial trophoblast, which is a bunch of fetal trophoblast cells fused together to form a barrier between maternal and fetal bloodstreams. So this is a really neat example. But then if we look in the genome, we can see that there's probably on the order of 100 transposon-derived proteins that show evolutionary signatures of constraint, which implies that they're actually functional. But by and large, we don't know what they do. And this is something that, another thing that my lab studies. We try to understand some of these domesticated proteins and their function, but I won't talk about that too much today. I should mention that, there's maybe about 100 transposon-derived open reading frames in our genome. That's pretty interesting. But by and large, I mentioned that most transposon proteins have suffered mutations such that they can no longer make a protein. And that kind of makes sense that we would have selection against these parasitic elements. So if we think about ERVs again, or endogenous retroviruses, there's about 400,000 fragments in our genome. But they initially come in like this, a full provirus. But most of them have either completely lost this middle part through recombination or have suffered protein coding mutations. And so this fact has been known for many years, and because of that, the research community has largely ignored ERVs and transposons, assuming that these are largely inert or non-functional elements in the genome. But over the past 10 to 15 years, there's been a renaissance in the transposon field, driven largely by the advent of whole genome assays for functional activity, such as RNA-seq, ChIP-seq, ATAC-seq, which many of you are familiar with. So with these unbiased data sets that give us these genome-wide maps of predicted enhancers and promoters, computational biologists have started to many came upon this unexpected discovery that about 15% of any enhancer data set that you look at, the elements have clear origins from within transposable elements. And so that implies that transposons, even though most of them no longer produce proteins, are a surprisingly abundant source of regulatory elements in our genome. So many studies have discovered this in many contexts, from healthy cells to disease cells to early development. Basically, any data set you look at, we see a signal of transposons. So this has been really interesting because this is actually harkens back to a very old hypothesis that transposons may actually be pivotal players in the evolution of regulatory networks. And indeed, this idea was put forth by Barbara McClintock when she first discovered transposons in corn. She hypothesized, before they were later found to be parasitic elements, she hypothesized that transposons were playing a role in development by controlling genes throughout developmental stages. And this is an attractive idea because when a transposon first invades a genome, it has some pre-evolved regulatory activity, but then it's copying and pasting that same sequence up to thousands or even millions of places in the genome. And if you're copying the same sequence with the same regulatory properties throughout the genome, that may be an evolutionarily fast way to construct or remodel a regulatory network where you have many distinct genes all under the same regulatory control. So this is kind of where our field is at. There's a lot of interest in the potential for these transposons to be driving our evolution in various ways. But where the key challenge we're at now is actually understanding the biological consequences of these elements. So we're able to see through ChIP-seq that a lot of binding sites come from transposons, but we don't actually have a good idea of whether that activity that we see is actually important or whether it's just a remnant of their old parasitic activity. And certainly both cases are plausible. All right, so I was interested in this challenge as a postdoc and decided to start working with regulatory networks underlying innate immunity as a model to study this idea. And I'll go over, very basics what we're looking at. I'm sure most of you are probably familiar with the interferon response, but for those of you who aren't, essentially when you're infected by a virus, your cells detect that and through various means and release a lot of danger signals into your bloodstream, including interferons. And here we're talking about interferon gamma, but there's many interferons. And when other cells detect interferon gamma through their interferon receptors on their cell surface, that activates a transcription factor, STAT1, also other factors, which then translates to the nucleus, binds to places in the genome, especially where there's certain motifs for this protein, and activates what we call interferon-stimulated genes, or ISGs, which are basically your front line of defense against infection or in sterile inflammation. A couple of features about this which really made it useful for us. One is that there were, given its broad importance in many fields, there are a wealth of genomic data sets that have profiled cells undergoing interferon stimulation in both a mouse and human. Another kind of unique attribute of this pathway is that we expect it to be under unique rapid evolutionary pressure because of its involvement at the interface with pathogens. So we know that, for instance, antiviral proteins are amongst the most rapidly evolving in our genome, so we presume that perhaps these regulatory networks may also be under pressure to evolve rapidly, and that might be a place where transposons could really play a role in remodeling this network. And finally, we're fundamentally interested in whether some of these transposons have a biological consequence, and innate immunity being many of the pathways can be assayed in vitro or in cell culture using FACS, and so that made it more easy for us to assay the functional consequences of some of these elements. Okay, so the first thing I did, or the first thing we wanted to look at, was whether the binding sites for STAT1 were shaped in any way by transposons in the human genome. And so we're kind of interested in this cartoon mock-up where, here we're seeing an ISG, and then we want to look at ChIP-seq data sets related to the interferon response. And we were interested to see whether some of the important binding sites were actually derived from ERVs or other transposons. And so what I did is I downloaded some public data sets produced by ENCODE and other labs, that perform ChIP-seq on STAT1 or IRF1 on cell lines treated with interferon. And for those of you familiar with ChIP-seq data sets, you often get basically a list of 10 to 50,000 binding sites. And essentially the first thing I did was ask, how many of those binding sites overlap with known transposons? And found that for all these data sets, about 15 to 20% of the binding sites have clearly land within transposons. So that gave us that first hint that transposons may indeed be involved in this network. I just want to mention that this is nothing special to STAT1 or IRF1. Generally, any of you who have ChIP-seq data sets, if you were to run this kind of analysis, would probably see similar numbers. And this is just speaking to the abundance of transposons in our genome. I next wanted to look at whether there were specific families of transposons that were showing a strong signature of activation. As you can imagine, I mentioned that there are on the order of 5 million transposon fragments in the genome. And so if you were to take any kind of data set, it may not be a surprise that there's going to be some overlap just due to random chance. And so what we were looking at specifically was for transposon families whose individual copies showed evidence of regulatory activity at a frequency that was greater than expected by chance. So we added some statistical measure to this, which I won't get into too much. But essentially, here's a kind of analysis that I did for HeLa cells, a STAT1 ChIP-seq data set. Each dot here, it represents a different transposon family, and each family can sort of be comprised of 10 to hundreds to thousands of copies of that family. On the x-axis is how many binding sites we expect based on the abundance of these transposons. You can imagine ALU elements, there's a million of them, so we would expect some baseline to be bound. On the y-axis, we see what we actually observe. And what we were interested in were the families that showed a lot more observed binding over expected binding, whereas transposon families that are along the diagonal are not ostensibly doing anything that special. So in this case, we identified across other cell lines, about 20 different families that showed really robust family-level activation in response to interferon. And by activation, at this point, I'm just saying binding of STAT1 based on ChIP-seq. Okay, so I'm highlighting here MER41, B, and E, and for simplicity, we're just going to kind of lump them together. And so we were interested in following up this family. Why is this family turning on in response to interferon, and what might that be doing for our cells? So first we were interested, okay, so what's MER41? Where does that come from? A lot of transposons have names like these. They're kind of cryptic. But basically, we looked at MER41 in a panel of different mammals, and we infer that MER41 invaded our primate ancestors about 50 million years ago. And that's because if we look in the genomes of rhesus, marmoset, human, and chimp, we all have MER41, and they're all in roughly the same space. So it probably invaded at a certain point, amplified, and then like all transposons, eventually went extinct through mutations. There are no such elements in mouse or any other non-primate species. And if we look at the LTR, or the original viral promoter sequence, it turns out that there's a pair of perfect matches to the known STAT1 motif in this LTR. So that's consistent with why we see so much binding of STAT1 to this element. I want to show some ChIP-seq analysis of this. So in this slide, we took primary macrophages that were treated with interferon gamma, and we're looking at both untreated and treated data sets. So here, in these heat maps, I've lined up 150 MER41 elements. So each pixel row is an element, and then each column is a 8 kb window of ChIP signal around that element. So, and we're looking at ChIP for STAT1 and H3K27 acetylation, which is a histone modification predictive of enhancer activity. And so we can see in untreated cells, it doesn't look like anything. It seems like these MER41s are largely inactive, perhaps junk DNA. But now when we see upon the same data sets upon interferon stimulation, these elements seem to coordinately wake up and have binding of STAT1 and other marks of enhancer activity. And so if you were to show this to kind of your standard researcher who's interested in gene regulation, they would say this looks like an enhancer to me. So based on the epigenetic marks, this definitely looks like a potential enhancer activity. Now, of course, we were interested in whether or not these had any biological function. It's entirely possible and probably true that most of these elements are not near any important gene at all. Many of these elements are kind of in the middle of nowhere, and that might be the reason why they've been allowed to persist in our genomes as mostly neutral mutations. But nonetheless, we were curious, and we started looking at each of these elements one by one and asking whether there were any known interferon-stimulated genes nearby. I'll go into one example right here. So one that we found that we were really excited about was this gene called absent melanoma 2, or AIM2. Some of you may have heard of this gene. It's important for initiating the inflammasome signaling response in response to cytoplasmic double-strand DNA. And so it's important for responding to DNA viruses, and it's also important in many kinds of diseases as well. So here I'm showing this gene, and it's pointing in this direction. And just 200 base pairs upstream of this gene is one of those MER41 endogenous retroviruses, which no one had noticed before. And if we overlay that ChIP-seq signals from various data sets, we see pretty convincing evidence that this MER41 element is showing interferon-inducible epigenetic activity. So you can see in blue is untreated cells, there's not much going on. In red is the treated cells, and we see binding of STAT1 and H2K27. Here I just thought for fun I'd show this conservation track, which is basically demonstrating that this element is present in the primates, but not in your more distant primates or rodents. And I should mention that AIM2, the full name is actually interferon-inducible AIM2. And so it's a known interferon-inducible gene, which is why it was a candidate for us. Okay, so then, at the time of doing this, we were really excited about the availability of CRISPR, which would now allow us to test the hypothesis that MER41 element was required for the expression of the gene nearby. And so I conducted that experiment. We took HeLa cells and designed guide RNAs that were located at the boundaries of this particular element. So that also included unique sequences on either side. So we were just clipping out this element and not one of the hundreds of other elements nearby, and generated multiple clonal lines where there was a homozygous deletion for this element. And then indeed, then we treated both control lines and mutant lines with interferon to ask whether AIM2 expression was ablated. And indeed, we found that. So here's a Western blot for AIM2 expression. Untreated cells, AIM2 is not on. In treated cells, it's on. But now in our CRISPR mutant, AIM2 does not turn on in response to interferon. Even though we didn't do anything to the gene, we just deleted the viral element nearby. So that was a very exciting, clean result for us. We want to take it a step further. We wanted to ask whether this actually played a real role in a viral infection, not just when we saturate the cells with a recombinant interferon. So other members in the lab fortuitously worked on a virus known as vaccinia virus, which is a model pox virus. It's a large DNA virus, which is known to stimulate the AIM2 inflammasome. And so we essentially treated our control and MER41 mutant cells, or we infected these cells with vaccinia virus, and assayed for inflammasome activity, which can be measured by essentially cleavage of pro-caspase 1 into its active form caspase 1, which then initiates cleavage of interleukin and pyroptosis. And so basically did a Western blot again to assay that. And so in wild-type cells, we see in uninfected cells, there's no cleaved caspase. In infected cells, there's cleaved caspase. Because this is a DNA sensor, when we rescue the cells, this AIM2 can recognize its own plasmid. But basically, we can rescue it, and then in our mutant cells, we see that this response is all but diminished, and there's probably some redundant pathways that are still activating this. But we can see that there's definitely a severe impairment to the inflammatory response to viral infection. So this really was a nice result that convinced us that this was an ancient retroviral element that had truly been co-opted and was now important for infection by a modern-day virus. And we thought that was a very interesting twist to what we think of as a host virus coevolutionary arms race. All right, so anything interesting in evolution can happen at least once. We wanted to see whether this was part of a wider phenomenon, namely, were there other genes also regulated by MER41, not just this AIM2 gene? And so we looked at there are many other genes, but we found 3 that were particularly good candidates: APOL1, IFI6, SECTM1, all genes that had previous studies by other labs implicating them in various immune responses. All of them also had MER41 elements bound by STAT1 nearby. In these cases, they were a little bit further away. They weren't the only binding site, But in these cases, I repeated the experiment, generated CRISPR lines for each, deleting out each of these specific elements, and then conducted qPCR to look at how that might have affected interferon-inducible expression of that gene. And so here are the results, and we can see in all of these cases, the interferon-inducible expression of the nearby gene was significantly reduced. We can see curiously that it wasn't the black and white 100 to 0 reduction we saw with AIM2, and we think that has to do with the fact that it's not the only STAT1 binding site in each of these cases, and that just might speak to more, or diverse ranges of regulation of some of these genes. Okay, so to wrap up this story, we then collaborated with another lab, and we were interested again in the extent of how much these MER41 elements really matter for our interferon response. And we didn't want to, making these CRISPR mutants is far easier said than done. It did a lot of cell culture work. Didn't want to do that for 100 more elements. And so we took a different approach, which was using essentially chromatin conformation assays. And I won't go into the details here. Here we used a bait that was specific to the transposon. So we made some tweaks, but essentially the take-home message is that we found a lot of MER41 elements with evidence that they were looping over to an interferon-stimulated gene nearby. And we found, I think in this case, about 200 of these elements. So we really think that this MER41 has played a major role in our own evolution of our interferon responses. And essentially here's our little model diagram. We have ERVs that don't do anything, but now they're important for the interferon response. And I think the implication here is that this may actually be an important mechanism that is driving the evolution of our responses in response to various pathogens infecting us. And this may actually drive species differences in our responses as well. I think that's an important point in that we often think of the innate immune response, cytokine responses as really basic conserved pathways. But now we're starting to see evidence that the very regulatory network that controls these pathways may be very different across species. Right? I talked about MER41, but that's only in primates, and there's no MER41 in mice. Right? So what does that mean for mouse interferon regulation? That's something I won't be talking about too much today, but basically in the lab, we're taking cells from mice and other species, essentially conducting the similar experiment, doing omics on the interferon response, and asking whether the transposons in those species have been co-opted in parallel to kind of do a similar thing. So I think, and preliminary evidence points to indeed that being the case. And so this really suggests that transposons may be this underappreciated but really important driver of divergence of these critical pathways in human and also animal models. All right, so I want to switch gears a little bit here. So that was published work that I did in my postdoc looking at the evolution of our immune responses. But when I started at Boulder about a year and a half ago, I became really interested in whether these elements may also play a role in pathogenesis. And this is something that hadn't really been looked at in the context of transposon regulatory activity. It, of course, has been looked at in the context of mutagenic activity. And I just mentioned this is a work led by a postdoc at my Ivanchevich. Okay, so our question coming in was whether ERVs may promote the evolution of disease-associated regulatory networks. And so we're all familiar with pictures like this. So this is TCGA data looking at different non-tumor and tumor samples. Each row is a different differentially methylated loci. We can see in cancer cells you have total genome-wide demethylation, you have total genome-wide remodeling of your epigenetic landscape, hugely messed up gene expression programs, and understanding the mechanisms, how and why that happens is a major goal in cancer biology. And so it's been known for many decades that ERVs and other transposons do become reactivated to some extent in many tumors, likely due to just their mutations in upstream epigenetic proteins and the like. So this, and I should say, the very discovery of endogenous retroviruses was due to its overexpression in tumors. And so this has led to decades of study into whether the reactivation of transposons is merely a consequence of the cancer, or whether there may actually be some kind of causal or at least a pathogenic effect of transposon regulation, or of transposon reactivation. Again, most of these studies focusing on transposon-derived proteins or transcripts, the stuff that we can measure by microscopy or qPCR. And I should mention that the results here have actually been pretty controversial, right? So some of these retroviruses have been called rumor viruses in humans because there's been a lot of controversial links between various retroviruses and breast cancer, et cetera. And I just want to make a point here in that it is now well established that retroviruses, specifically endogenous retroviruses, can cause cancer in many animals, including mice, koalas. It's actually leading to koala extinction, deer, pigs. And that's because, and through the mechanism where the retrovirus creates a new insertion and causes an oncogenic mutation. So that's a known mechanism. But in humans, that's not the case, because in humans, for whatever reason, no retroviruses are currently active anymore. And so for that reason, the activity of retroviruses in human cancers has largely been looked at to be sort of a correlate or a biomarker of cancer, but not necessarily having any kind of pathological effect. But our hypothesis was that perhaps it's not the proteins and transcripts we should be looking at, but maybe the epigenetic or regulatory activity. So these could be being reactivated, but the reactivation may cause dysregulation of a nearby cellular gene, and that dysregulation may be a pathogenic a consequence of ERV reactivation. So this is the idea that we started to chase. And one of the reasons why that hasn't been looked at is that there haven't really been the relevant data sets out there profiling cancer tissues for their non-coding regulatory elements. So looking at these elements requires you doing something like ChIP-seq or ATAC-seq. But now, over the past couple of years, these data sets have become more and more widely available, and now there's just going to be a tidal wave of these types of data sets. It's a really good time to investigate this hypothesis. So for our first pass, we took a data set from the Scacciari lab where they took colorectal cancer. They were basically investigating colorectal cancer and looking at the enhancer landscape by conducting ChIP-seq for H3K27 acetylation. They had 7 tissues from healthy patients and then 36 samples, which were a mix of patient-derived cell lines and primary tumors of colorectal cancer. And again, we were interested, so we downloaded this data and reanalyzed it, specifically looking for, again, transposon families that were enriched in these data sets, similar to what I had before. So essentially, we're kind of trying to see whether there was a MER41-like thing, but for colon cancer instead of for the interferon response. And I should mention that we used a really cool tool called Giggle, which is developed by one of my colleagues at BioFrontiers, Ryan Layer. And this is a really neat tool which I think should be useful for a lot of cancer biologists, which allows you to take the data sets that you generate in your lab and query them against the universe of data sets that have been published already out there in GEO and other public data sets. So here again, we were interested in the families that showed, we wanted to find transposon families that showed recurrent and strong activation in cancers, but not in normal cells. So here's a volcano plot depicting this analysis where each dot, again, is a transposon family. Dots on the right show strong enrichment. Dots on the left show a depletion. We weren't so interested in these, but we were really interested in the ones that showed enrichment in these colon cancer samples. And what we found was this family of LTR10, ACNF. Again, I'm just going to kind of lump them together for the purpose of this talk. And what we found was that these LTR10 elements derive from a different retrovirus. So, there's about 400 different ERV families in our genome. This is yet another one, and this is called HERV-I. And this is interesting because no one really had published on this. HERV-I is a largely neglected element in our genome, and part of that is that it really isn't active in many tissues in a healthy individual. It seems to only become reactivated in this cancer state. Again, looking at its genomic distribution across different species, we infer that this one came a little later. This one came about 30 million years ago. So this one's not present in marmoset, but it's present in our closer primate relatives. And again, it invaded and distributed about 1,000 copies throughout our genome. Okay, so now I want to show a specific element that's in our genome. So here's a region of chromosome 2 where we have a full-length HERV-I element. And by full length, I mean that you have the LTRs, and you also have the viral proteins in the middle. And here, the tracks I'm showing H3K27 acetylation ChIP-seq from that same colon cancer data set. The first 5 lines are control tissues from healthy individuals, and we can see that there's not really any activity. But then in the bottom half, we see each of these samples shows evidence of regulatory activity at this element. So here's one element. And then now I want to zoom out again to show a heat map of where now we're stacking Each pixel row is a different LTR10 element, and we're looking at K27 acetylation in a window of 4 kb. On the left columns are 4 normal samples, and on the right columns are 4 cancer samples. And we can see again that this cancer-specific activity, or I should say epigenetic activity of this mark, appears to be quite consistent. It appears to be, and again, all these rows are matched, and so the same elements that are silenced are activated in the cancer samples. Another point is that not all of them are activated, just about a third or a half, and we're looking into why that is. And part of that could be chromatin context, or the fact that some of these elements may have suffered additional mutations or deletions that make them unable to bind this, or unable to activate as an enhancer. So this was our initial analysis based on sort of a small data set from one study. But then while we were doing this, the Howard Chang's lab from Stanford published a huge ATAC-seq data set of TCGA samples. So they conducted ATAC-seq on many patient samples, and we decided to pool from that and get colon cancer samples to see whether these elements also show accessibility in a larger cohort of patients. And so, I don't expect you to squint too hard on this, but this is basically that same heat map, but with ATAC data, looking over 80 individuals. And we can see, and these again are LTR10 elements. And so we can see again that these LTR10 elements, again appear to show this epigenetic reactivation in most, if not all, colorectal cancer samples. And I personally was actually very surprised to see so many of these samples show this signature, simply because, there's many subtypes of cancer, and there's, you don't expect them to all show the same molecular signature, but we do see a very striking recurrence of LCR10 reactivation in cancer. Our next question was why this was happening. And we're still digging into this, but we have some preliminary ideas. Essentially what we did was a motif analysis. So this is a very common analysis in genomics where you just have a bunch of elements and you want to ask what sequences are overrepresented here. It's a little tricky to do that with transposons, because if your input were just transposons, you would get the entire sequence as a motif. So here we actually took LTR10 elements that showed epigenetic activation and compared them against the ones that didn't. And we came up with this motif, which turns out to be the motif for June B fossil, or essentially components of the AP1 complex. And that was interesting to us because the AP1 complex is often aberrantly regulated in many tumor cells. And it turns out, again, if we look at the original LTR consensus sequence of the original virus when it first invaded our ancestors, there's a trio of these binding sites in this LTR. So consistent with the idea that this might be binding these AP1 complexes. We then looked to ChIP-seq data that was produced by ENCODE, which conducted ChIP-seq on June and fossil, which are AP1 components, in HCT116 cells, which is a colorectal cancer cell line model, and found that indeed the LTR10 elements that are active also show binding, based on ChIP-seq of this element. So this is kind of giving us a hint that these elements are indeed being bound somehow by AP1. We still don't understand whether AP1 is necessarily off in healthy cells or whether there's some additional de-repression or other mechanisms that is now allowing AP1 to bind to these elements and potentially activate their enhancer activity. All right, so finally, of course, the real question is, we see reactivation of these ERVs throughout the genome. There's probably many other families that have the same story, but does this reactivation have any effect, A, on cancer gene expression, but also B, on cancer cell function? So this is ongoing work. We don't have many answers yet, but again, we were interested in looking for candidate genes, so we looked through each of these LTR10 elements, and we were looking for genes that, A, had RNA-seq evidence of expression in colon cancer, either cell lines or tumors, and also had some kind of previous literature association with colon cancer. And one candidate that we started looking at is this ATG12, which is an autophagy-related gene. So that's the gene right here. And then way far away, about 70 kb upstream, there's the pair of LTR10 elements, which have, again, really strong enhancer activity in tumor cells, but not so much in healthy cells. And so this is an element we're interested in testing. We've done some preliminary studies using CRISPR-I. And so this is not a true CRISPR knockout, but it's a little bit faster, where we've designed guides to deliver a dead Cas9 fuse with the crab to silence the enhancer rather than cut it out. And we're starting to see some results. So here, this is qPCR of the ATG12 gene. In a control, we just targeted CRISPR-I to GFP, so it shouldn't do anything. As a positive control, we targeted to the TSS of the gene. And we don't see a huge reduction, but we do see reproducible, consistent reduction. We still have to work out this CRISPR-I protocol. It's not perfect. But what surprised us is that by silencing that far distal LTR10 element, we see the same amount of reduction as we see if we silence the promoter. And so we're pretty excited by this preliminary result. But of course, our next step is to actually make a true CRISPR knockout of that element to see the effect. We want to do a few more knockouts on other candidates and also other colorectal cell lines just to make sure this isn't just a sort of specific thing to these cells. But moving forward, something I'm interested in getting your feedback and help on is looking bigger and asking, A, what is the mechanism? I kind of alluded to that for the activation of these LTR10 elements. I think that's critical because we often think of transcription factor complexes such as AP1 as having a major pathological effect on a tumor cell. But it would be very informative and important to know if a lot of that action was mediated through their binding at some of these transposon elements. So that's something we're interested in examining. And then another important thing in terms of understanding their function is to go beyond gene expression, right? So I'm pretty confident that we'll be able to knock out these elements and see a difference in the expression of the gene. But whether that has any consequence on cancer phenotypes of that cell, either in vitro or in some kind of a xenograft experiment, that's going to be the real question to answer. And so, that'll be ongoing in the next year or so. To kind of to wrap up or expand our analysis, everything I've talked about today was really about colon cancer. So I should say, I took the ATAC-seq data set from TCGA, and that was ATAC-seq done on many of the cancers, not just colon cancer. And we asked about LTR10 and other transposons, whether we see reactivation in these other tumors. And indeed we do, and one thing we found was that different tumors have different reactivated ERVs. So there's some heterogeneity here. So here we find LTR10 really lit up in colon cancers, but if we were to say, look at uterine endometrial cancer, it's a different set of transposons. So this is really speaking to a very complex pattern of reactivation that we're very interested in following up in the future. All right, so to wrap up, hopefully what I've convinced you today is that ERVs in our genome are really a pervasive source and until now, underappreciated source of regulatory elements, but they can have both beneficial and potentially pathological consequences on our biology, and I'm interested in studying both of these. So, at this point, all we can say now is that we think indeed these are a pervasive source of cryptic enhancers in various tumors, but now we're interested in understanding their function. We want to know whether their reactivation has true pathological consequences. We want to understand why certain ERVs are reactivated in colon cancer, but not in other cancers, and vice versa. And finally, again, getting back to our evolutionary interests, is that if it turns out to be that ERVs are responsible for determining, say, the cancer gene network of colon cancer and other cancers, that has significant implications for understanding or studying these cancers in animal models, right? So if the gene network of colon cancer in human is in part dictated by primate-specific retroviruses, then when we study that same cancer in mice, we got to be careful about thinking about whether genes are regulated the same way. And so I think that has implications for interpreting animal models, but also I think is an evolutionarily interesting concept in that many of these elements may be spreading, may be virtually neutral because they're generally silenced by our epigenetics, but may actually have a major role in contributing to the evolution of our disease networks, which can then affect our biology now, even though these viruses invaded us over 30 million years ago. So that wraps it up. I want to thank the members of my lab at Boulder. I want to thank the funding, which comes from NIH, the Betcher, and the Sloan Research Fellowship. Thank you for your time. Yeah.
So, Ed, that's very fascinating, really cool. So there's two sort of ways to envision it. So you mentioned that, I know it's the mouse AIM2 is also interferon inducible, but maybe through a completely different mechanism. So one idea was that their last common ancestor was not interferon inducible, but I would actually favor that it was, and that what happened in the lineage that ended with us and chimps and such is that retroviral element within there essentially created redundancy. And so whatever used to be the interferon regulatable element of AIM2 is no longer. And for those other genes, they haven't lost their other mechanisms, so they still maintain interferon regulatability, if that's a word, despite the fact that they now got this element.
Yeah.
They may be destined to lose that redundancy at some point.
Yeah, that's a fantastic point, and certainly along the lines of what I'm thinking. So first, mouse has AIM2, other species have AIM2, but I would say it's actually kind of controversial whether AIM2 is interferon induced in mice. I think it's, in many cell types it's actually constitutive. So in that specific case, it's unclear. I actually think so. In mouse, there's another protein, IFI-202, which acts as an inhibitor. So I think they've evolved a separate mechanism to dampen its activity. That being said, that's AIM2. I think that's a really interesting and oddball case. But your overall point about new transposons essentially acting as duplications of regulatory elements, then lessening selective pressure for that original element, that probably happens a lot. And so we think that there's going to be a lot of cases where if we were to profile the same regulatory network in a panel of different animals, the same gene may be regulated by different transposons in every single case, but it certainly could have been the case that the overall expression pattern has been maintained and conserved the whole time, and it's just simply this kind of nearly invisible turnover that's happening upstream. That's very interesting, something we're definitely trying to check out. I think that is a major mechanism. Yeah?
I've got two questions. One, a specific one, and one kind of general. So the specific question is, colorectal, one of the advantages of studying colorectal cancer, it goes through this very defined progression from normal to pre-neoplastic to neoplastic to malignant.
Huh?
And so it would be interesting to look and see where LTR10 turns on, where in that progression
yes. Very interesting, yeah.
And I presume you just use cell lines, which are usually...
Yeah, cell lines or tumors, but we have, it would be amazing to have that kind of a time course, yeah.
So that would be an interesting thing to see where that turns on. More general question is, you've been talking mainly about protein encoding genes. Have you looked at all on non-coding RNA, microRNAs, long RNA?
Yes. Yeah, we have. So that's a great question. It turns out that most non-coding RNA is actually derived from transposons. So, anyone who studies non-coding RNAs knows is controversy in the field how many of them are actually functional versus kind of spurious transcription products from the genome. And actually kind of favor the latter in that as these, especially ERVs, deposit in the genome and they lose their protein coding capacity, a lot of them maintain that LTR, and that LTR is a viral promoter and has a transcriptional start site. And so I think that in humans at least, about 20 to 30% of all non-coding RNAs actually initiate from a ERV element. And so, and it's oftentimes the LTR initiates maybe the first exon and then splices into some cryptic splice site into the genome. And you're basically just transcribing this random gene. And, many times that will just simply decay away, but sometimes may be co-opted into a real functional non-coding RNA. So in the case of MER41, we're actually studying one. Some of you may have heard of a non-coding RNA named Banker, which is a marker of melanoma, discovered by Paul Cavare's lab. And this Banker non-coding RNA, stands for BRAF activated non-coding RNA, is actually almost entirely basically a MER41 element that has lost its protein coding capacity but is now transcribed as a link RNA. So we're looking into that one, and we're CRISPRing it out and trying to see what it does. We haven't looked at that for this cancer case, this LTR10, but other people have. So I've been mostly focusing on transposons as a source of non-coding enhancers, but in the past 2 or 3 years, there's been huge papers out looking at TCGA RNA-seq data for the contribution of transposons, both to non-coding RNAs and also as alternative promoters to protein-coding genes. And it does indeed appear to be they are indeed a major source of that, but we think that's the tip of the iceberg. Most of these ERVs have lost their promoter activity. It can now only function as as distal enhancers. So we think there may be more activity as enhancers. Yeah.
So that's a very interesting talk. So you brought up the question of tumor heterogeneity.
Yes.
The big question is, I mean, within the same tumor, is this reactivation is it consistent throughout, or is it something that starts at the tumor initiation phase and then maintains itself, or depending on the tumor microenvironment, you have sort of deactivation, activation, depending on the sort of the dynamics of the microenvironment?
Yeah, that's a super interesting question. That's something that where we should go. Yeah, again, all this is in bulk, and so we don't know. I think every possibility that you mentioned probably is true. You know, I think in your initiating tumor, there's probably a few transposon families that are de-repressed, and that de-repression is likely to continue on in its progeny. But also, as the tumor grows and develops, you know, there's probably going to be some level of heterogeneity, even within the same tumor cell populations, of which ERVs are kind of on or off. And we actually think that ERVs and other transposons may even be a really likely candidate for driving heterogeneity, because unlike normal enhancers, transposons are dually targeted by both silencing and activating machinery, right? We have transposon silencing machinery, but the transposons themselves are trying to hijack the cell machinery. And so I feel like this tug of war makes it more likely that any individual element which shows kind of ultimately stochastic patterns of activation or repression across cells. So we're very interested in eventually going to single cell technologies and imaging to try to investigate that question.
Great, let's thank Ed again.
Thank you.